Welcome everybody. Yeah, really happy to have you in this two-day tutorial on Sfaira. We have like a ton of material prepared, like sort of tutorial style presentations of parts of the API, and then more like lecture style mini presentations of other parts. We'll kick off today with a short introduction to Sfaira by Fabian, who's gonna take over the screen share now. So welcome, Fabian.
Yeah, thanks everyone for joining to our Sfaira workshop. This is really exciting to have so many people be interested joining here and potentially quickly showing their video and waving their hand to have a little bit of a workshop conference feeling. Not sure you really want to go for that, but it might be nice. I'm not sure. I guess at some point we do a little bit of a yeah, excellent. Thanks so much. Feels a bit more connected, right? And I hope we actually see each other this summer. And there's a whole bunch of meetings coming up, which will be a big opportunity. I think it will be fun to have a little bit of a survey or so where people from, what backgrounds are. And, at the end of this, we will be doing a short thing where we'll be asking you, what you learned, if you think we should be doing this again, or we should be adapting this and so on. You saw this is hosted from our single cell omics network Germany. This is a sort of a more local speaking thing that we've been setting up. Where we wanted to educate people in this direction, and this is sort of our one of our activities beside the bigger conferences and regular talks. I just spend something like 10 minutes on introducing a little bit what the outline of this thing is, and then hand over to David, Leander, and Felix, who can carry on the workshop and actually really the main force behind it. Really happy and thankful to the three of you that you've been doing this already now. So briefly, my name is Fabian Theis. I'm from Helmholtz, Munich, and the Technical University of Munich, an associate position at Wellcome Sanger, interested in single-cell genomics methods and computation analysis for a long time. I guess most of you have seen that single-cell genomics is becoming big data. Well, why? Because, for example, we have these droplet-based assays, and so many people are using this. And this is has been named Method of the Year in all its variants, in normal, multimodal, spatial, and data sets are exploding. So I think many of you have seen this, and mostly what we derive from this, at least how I usually use this intro slide, is saying, yeah, since we have these big data sets, well, kind of tells that we should be using some type of AI machine learning tool to then do cool things with this big data. And that's of course true, and I will show you a little bit about that in a minute. But I think one thing that might be a bit under thought for, or maybe not as popular, because it's just working, you don't maybe so quickly make an analysis of the paper, is the data management aspect, right? So because these things are becoming so much, and I think nowadays, in pretty much all the papers, we're expected to reflect our own data set with respect to others, our own methods with respect to others. We need to be able to somehow manage and handle these data quickly and sort of put them into context. And even let alone the public data sets, which is something in part that we do with Sfaira. Also your own in-house ones. I guess in bigger labs, it can become quite many quite quickly. And I think it's useful also to have some means of organizing in this way. And this is, I think, something that has been sort of at the brunt of when we set up this Sfaira method. Let me say a little bit about what the underlying sort of architecture and well, not architecture, but maybe sort of vision with respect to data integration is. For this, starting briefly with a competition that Malte from the lab, together with a whole bunch of collaborating labs, has been set up as this multimodal single-cell data integration challenge at the recent NeurIPS workshop, where we've been looking at 160K cells across RNA, attack, and protein, and had this cross-modality prediction. And I won't be saying something about all of these methods, but the key take-home I got from this competition was pretty much all of the winning models used some way or another a neural network. Just I guess because scales are high, maybe also because it's been a bit biased because it's been presented at the machine learning conference, so people like that. But I think it tells us that we're sort of entering this regime that computer vision has been doing for quite some time, that they have been always dealing with bigger data sets, and then also with these methods to analyze that. And I should say this data set, which is I think a beautiful I think it's one of the largest, maybe the largest multimodal single multi-site data set that's around. This is in bone marrow. It's available also as a data loader via Sfaira if you want to check that out and play around with it. So yeah, this is one of these architectures that we often use, and it's interesting because this bottleneck layer sort of tells you something about it, and we have some interpretation of that. Won't really say much about these networks, but the one point I'm trying to make is that we know how to reuse some of these structures. And that might not be as straightforward with, let's say, a single cell KNN graph or so, where we had to do some rather more elaborate operations to make these map on top of each other. But this one is rather simple to do the data integration challenge. So lo and behold, I think many of us would agree that, when you first set up the human cell atlas, we just had all these parallel atlases. But I think nowadays we're entering this regime where we want to reuse these reference atlases. That's why we want to have existing data sets compiled, ideally served via HCA or HubMap or other consortia, and then mapping our own data on top of that. And whenever we need to easily use those. And we've been doing this in some settings in big consortia, for example, to query a variation of entry genes of SARS-CoV-2 and across a whole bunch of the lung atlas teams. And there's challenges. How do we map on top of it? How do we deal with these distributed reference data sets, and how do we make those maps accessible? And this is from a beautiful paper from Raoul's lab, where he has been talking about sort of querying your own data set on top of a reference, and then doing integration and prediction. And I think this part here, this querying on the reference, sort of serving this reference, is something that I think we try to sort of cater to with the Sfaira tool that I mentioned a minute, and that you could potentially today learn how to contribute and maybe also sort of put in your own data set there. Last concept point that I wanted to make, I'm not sure you saw that, a beautiful paper from one of the developers of Rosetta, of the AlphaFold competitor, and Peter Sorger and Nature Methods last year, which they call differential biology, where they said, now that you have these deep learning models, you could actually reuse them to maybe model molecular mechanisms. There's all kind of architectures that you can build in mechanisms, such as spatial invariants and so on. And I think putting these typically data-driven methods that we mostly do together with this a priori information, such as, we're doing, I think many of you have seen this RNA velocity, right, where we sort of relate unspliced and spliced reads and learn something about a bit more mechanistic. Putting all of this together under one roof, I think is exciting. By the way, in Sfaira, you can also now handle unspliced reads. That would just told me. In any case, sort of putting things together and being able to concatenate models necessarily means that we need to share data and then also recycle models in order to be able to put them together. Computer vision is doing this, of course, and we just had a speaker from Google Cloud here who's basically offering each of these models as a separate fee-for-service type of thing. Maybe not that far yet in our field, but I think this will come soon. So how can we reuse these primitives? One thing that we've been thinking about, for example, is transfer learning. You have a data set, you have one task, you have another one, and this would be traditional. And transfer learning, you sort of transfer this knowledge. And we came up with one method to sort of map your own little data set here on top of this much bigger reference one, which uses an operation on the single cell graphs. So much for reusing models. What we have in Sfaira is then this single cell data and model zoo that essentially tries to really annotate, compare, reproduce, and then put these data sets together. And so it has not only data sets and as well as annotation, but also models and parameters. Today, we'll be just focusing on this part. But of course, there's more to it. It has been originally developed by David and Leander. And quite recently, we're happy to say that we hired a new PhD, aptly named also Fischer. But this is sort of the D, this is the F version now. And very happy about Felix also contributing to that and be around at this workshop. So this is Sfaira. Again, I won't say much about the architecture, particularly because I think that would much better be explaining it. But the key part is that we have a dataset API, how to facilitate data reuse without you actually sort of needing to serve this from a central location. Essentially, for each of these published data sets, you have a loader that will tell you where to get it from HCA, from CZI, whatever, right? Or you can also have it for your own private data set. Later, there will be a session dedicated to that, how you input your own data set without actually needing to share it to the world, putting it to some public server, but still being able to deal with all these public models. Then we have something about those learned models as well as those parameters. Let's ignore this for now. Let's really focus on those curated data sets, which I think is a major use case for many people. So the aim today is really how to learn, how to write, how to use this file, and how to write maybe also your own data in order to put your own data set into this thing. All right, Sfaira, and this is a bit of an older slide already. I think at the time, like half a year ago, so we're kind of serving something like 150 data sets across 55 organs and more than 8 million cells. David just told me it's more than 10 million potentially now. But what's kind of neat, because of this sort of extended architecture, that can easily not just take like one data set from one lab, but maybe we take all the blood data sets from human, or you see how many different ones there are in, or maybe just take like a whole human set of organs and stream them into in one big data file, and then sort of doing something downstream with it. So I think that's kind of useful. You can of course then analyze metadata for it. You can sort of check out how how this actually works, and you'll be doing this in a minute. And I think that's an interesting feature that was just recently added that you might not have seen, is that in a newer version of it, we're actually interfacing with CZI cell by gene. So Sfaira, I should mention, has actually gotten some very nice generous funding from CZI, and we've been interacting with them on various aspects, but in particular this intersection of this fantastic visualization and a much more extended scope of cell by gene, which I think developers from cell by gene will later potentially be able to tell us about. It really helps us sort of use curation that might happen on both sides, but particularly on the Sfaira side, and push it in cell by gene side and vice versa. So I think we have essentially APIs to connect these two ecosystems. And then the last thing that I wanted to say is that you can, now that you've been trained such a data loader, potentially you can really replace some of these tedious workflows that you keep doing again and again for new data sets to be faster. Let's really imagine this day where, big vision, at some point we are able to really use single cell RNA-seq or some maybe more efficient, cost-efficient version of that in clinics. We don't really want to redo this every time, right? We need to really just jump from this process that typically has some analyst bias, sample speed, thoughts, maybe pre-learn embedding, where you could then have a speed up from potentially even days or even longer to minutes. And this is an example how we've been doing this for PBMCs, where here colors would be indicating different subcell types, which you either do with a manual embedding and annotate, or you take a Sfaira embedding, and here you will be just using the manual annotation from this one, and you take this Sfaira annotation, and they kind of overlap very nice, and you sort of get this for free by this efficient process here. And that has been useful already for us in a whole bunch of examples. You saw this maybe big data benchmark that Walter was been running, where he's been using some of this backend code, or Leander at an ICML workshop 1.5 years ago, where he's been sort of just quickly validating a whole bunch of methods on many public data sets. So with this, let me wrap up. This is the plan for today. I think David will throw this up in a minute. Before I show the plan again, this is the challenge. And I hope you saw my fantastic animation. Let me just replay this because you might have sort of gone into Zoom fatigue mode. This is a plan, but what I have as a challenge for you is everyone writing their own data loader. So our hope would be, if by the end we've been having like, 50, 80, maybe even more new data loaders for maybe your own fun data set that no one else maybe has been using, that would be cool. And you're gonna gain some single cell street cred by being a Sfaira curator, and to get like a mention on GitHub. Yeah, maybe that does it for you. This is the lab in maybe times when we're actually meeting each other. I really gratefully thank CZI for supporting the Single Cell Omics Network, BMBF for the support. And that's the schedule again. Really looking forward to see how this develops. And thanks everyone for this really fantastic interest, like so many people being interested in this, and hopefully helping us also to understand where maybe the needs are, what things are useful, which parts are not, and where this thing could really help you speed up your research and amplify results. We'd be really grateful to hear. Thanks.
Thank you, Fabian. Yeah, cool. So that was like a brief introduction to the entire project and like sort of like all of the auxiliary activities that are around Sfaira in our lab. Now, today we'll talk mostly about APIs. So sort of like explain like how we abstract certain operations that you may want to like execute on data sets. And the way we structured this is that we have sort of like 4 main API-centered sessions, 3 of which will have a tutorial after. So there is always like a block of us presenting briefly sort of like what the API looks like, and this is slide-based. And then afterwards, we'll go through a Jupyter notebook with some example code where you will, you could potentially like execute this notebook like alongside us, or you could just like watch us sort of like talk over it. So it's like up to you, but it's gonna be like more like sort of like real application case. In general, you can always ask questions in the chat or on Slack in the Fourier channel, and we have one person moderating the questions. So for now in this session, it's gonna be Leander. So yeah, just post them. He's either going to reply directly in chat, or he's just going to ask it. So yeah, don't feel like you need to wait until like the end of the session to ask questions. You can of course also ask at the end, but if you have something that relates to the slide that's up, just shoot. Yeah, then we just posted a feedback form on Slack, and I think we're also going to post it here in the chat. So if you want, keep that open and yeah, leave some comments like during the day today. Also, the agenda is there as well. All the times on the agenda sheet are rough. So it should be like around the anticipated length, but it may deviate a bit. So yeah. So if you log off and log back on, it may not be the case that we start a session exactly at the anticipated time. Then lastly, the last session for today, if you check out the agenda, is the open Q&A for tomorrow. So that is A, not mandatory, and B, also not structured. So if you feel like you're all set for tomorrow, you can essentially leave then. If you have any questions like installation issues, or you haven't managed to find a data set that you're interested in annotating, or you managed to install it, but like you're really lost in where you would like turn it on or something like that. Today in the last session, this is like really the time to ask that, because tomorrow we really want to like focus sort of like all of our communication capacity on helping you with metadata annotation, because that's really like complicated and there's a lot of stuff to learn there. So yeah, any of these issues, we have a lot of time today, sort of like roughly between 5 and 6. Just ask us then ideally, or write us an email tomorrow before the second day. Cool, I think that's the setup. Then let's briefly check out the exact sessions that we're going to cover. So having had this like bigger Sfaira overview of Fabian.
Hey, David?
Yeah.
So sorry for interruption.
Oh,
I was just wondering, because like you've been planning a whole big agenda. Do you like want to take some questions first? Is there people still asking about like how things are organized or where they should join? Is that sort of roughly clear for everyone? I mean, obviously you can ask in the chat, but maybe someone wants to really have a general question of interest or something.
Maybe one thing we should highlight, we'll have breakout rooms throughout the like all of this, but you only need to use them during the tutorials. So there's today there's always going to be one breakout room open, and you can go there and like ask sort of like, why is this cell not executing type of stuff. Tomorrow we'll have more of these breakout rooms to help you like more with metadata annotation. But yeah, so essentially you can stay in the main session today. Unless you have a pressing question in a tutorial, then you could just like jump in and out of the tutorial, the breakout rooms. But yeah, I mean, any other general questions, happy to take them now. I guess that's fine then. Cool. So the main sessions today are first, I go a bit more over the Sfaira project, but sort of like centered on the data API. Then the first API chunk-centric session is universe and dataset, which are words you hopefully be able to associate after that session. Then cellxgene interface as a second API chunk. The third API chunk is the store API. And then 4, also sort of an API chunk, is essentially strategies how you can manage private data with Sfaira, where private means that you may want to write a data loader, but you're not yet ready to share it because it's pre-acceptance or something like this in a publication. And then, as mentioned before, lastly, the optional Q&A. And yeah, brief outlook to tomorrow. So tomorrow is going to be the menu project. So tomorrow we go over how we curate, what curation really means, and then we'll walk you through curating one data set of your own. But yeah, we'll briefly go into this in the last session today again. And then at the end of tomorrow, we'll have an invited talk by a member of the cellxgene curation team, Jason, who's going to talk to us a bit about how you can use the created objects that come out of the end of the Sfaira creation pipeline to upload them to cellxgene, for example. So you may want to do that to sort of advertise your own data because there it's like really nicely presented. You may want to do that so that you just like share your creation efforts if it's not your own data. Yeah, but we'll go over that tomorrow as well. Okay, so then for the first session today, we go a bit about motivation of Sfaira data. So in principle, Sfaira, as Fabian mentioned, is largely a data API and a models API. And we bundle them in one package because there's strong synergies in streamlining data and models. So essentially the idea is, if you curated the data set well enough, so if you characterized well enough where particular parts are, like a count matrix, like a certain cell type and what that cell type is called, then it's easy to feed that data into a training for a cell type prediction algorithm, for example. Or if you have the expression data well curated, but you don't know cell types yet or other metadata that are predictable, you could easily execute a predictive model on that data. So they're really like, it's sort of like two things that go very hand in hand. Yeah, and then as Fabian presented, we have a bunch of application cases, like for example, cell type prediction for that. So we call it Sfaira because Sfaira is ancient Greek for globe or sphere. So here, like Sfaira, like sphere, it sounds sort of similar. And it's a code base for sharing interoperable data and pre-trained models. So there, the idea was really, we're not necessarily the only people who push new models, but rather like we provide like an interface for people to run their models. So an example would be in cell type predictor, right? It always takes an expression data and it puts out cell type predictions. So that's like a structured interface. And then you may want to choose different types of models for that. That doesn't really matter to us. But yeah, so the core idea is essentially, once you abstracted this interface, it becomes very interoperable. Similar for data, right? Like once you created data, they become very interoperable in the sense that everybody knows where to look for cell type annotation and everybody knows the rules based on which cell types are called a certain way. This was inspired by Kipi at the time, which is a framework in functional genomics. And they sort of underwent like a similar effort in like providing a streamlined interface for models in functional genomics. So we published this last year after having it published as preprint, I think the year before that, but we're still working on this code base of the Python package a lot. So this is really like under active development. And I guess especially last year, we put out a ton of new features. So improving existing features, but also adding new ones. So this is really like a living project. And so because this workshop is going to have like a bit of a data focus, we are at a point now where we really believe that we can make Sfaira sort of the dominant source of single cell data curation code because of the way sort of like we simplified this process, making it possible that we can scale it like within one person. So one of us can write like a lot of these loaders now in a very short time, but also it's very accessible to third parties. So like what hopefully we can convince you of tomorrow is that really also like people who have never really worked with Sfaira can curate data with it. Yeah, in a minute I go over why we really believe that this is going to be useful for the field. So yeah, right now we have I think almost 60 studies now and more than 10 million sets curated. So briefly, this data and model API, what does it mean? So the model API, it exploits the fact that data is structured. So it exploits the fact that you can sort of automatically say where parts are in a data object, like in an undata. And then it rearranges the input according to what a streamlined model wants. So it may want like these 5,000 genes, right? And then it may just like pull the corresponding features from a data object. And this is all controlled by our ontologies, which we're going to go into, other metadata structures. And the examples are cell type prediction, embedding, so reconstruction, but it could be anything where you can sort of like imagine an abstracted interface. And now the neat thing is, so from Theis we push our models to Zenodo. So there's a Zenodo with the parameters of our models up. And it's, so if you were to decide that you also want to push parameters to Zenodo, like that would be entirely equal. So there wouldn't be any hierarchy between our Zenodo and your Zenodo, like both are accessible by a user through the same interface. Now the data API, again, right, like you're pulling stuff from the cloud and it's sort of streamlined. Before it was parameters from Zenodo and maybe also code from other GitHubs. Now it's raw data files, like unprocessed count matrices or curated data objects. So they're really distributed over clouds, right? So like some people drop them on the supplement of a manuscript, so they're on the journal server. A lot of people put them on Geo. Then I don't know, some are on Fischer, Cell by Gene, Human Cell Atlas servers, or like institute-specific servers. There's a lot of these. Now the core motivation for having a data API is actually for to empower you to interact with this data that's like sort of like all over the internet. And then once you sort of like located it, being able to like work efficiently locally with it. So now this working efficiently with it, I call this the dark matter of single cell analysis code, which I described in this text, which you'll hopefully not be able to see, depending on your screen contrast, I think. So this dark matter of single cell analysis code, which is stuff like IO for files, maybe URLs of where you obtained a file from. In the best case, like often this is not even recorded. But it's also cell type matching code, definitions of batches, like biological batches. This is a bunch of code that exists in almost every single cell analysis project. That is often replicated, so it's redundant in the sense that a data set is published once to GEO, and then in every project, people would like write this code again, right? It would look slightly different because they would be looking at like maybe slightly different sets of metadata, but it would be very redundant about across projects. But because this code is streamlined so little, it was essentially very hard to like carry this over between projects, which is the reason why there's so much of this out there. So we think that this is like a huge liability because so for one, this is code that is often very hard to look at and is often not the key finding of a paper. So I think the number of cases where somebody else actually checks this code is very small. So this is bad, right? Because there may be bugs in there, and this is pretty important. Like if you mess up at the stage of data reading, which, I mean, it seems easy, right? But there's so many steps along the way that it could easily happen. If you mess up at that stage, then a lot of your downstream stuff is affected. So this is an issue for reproducibility, but it's also an issue for just essentially generating wrong results. On the other hand also, so imagine you didn't have to write that code, you'd be much faster at piloting projects, right? Because you'd have the data stuff like at your hands, and then you could just focus on writing a model, or you could focus on doing a differential analysis, something like this. So one of our missions is to organize this code, so this dark matter, into something like that is not redundant, that is more reproducible, and that is shareable. So that would look like this then. Essentially, before, your project is sort of like this code that nobody really wants to write and nobody really wants to look at, which is like the IO and streamlining code. And then the stuff that you generate manuscript figures from, so like the interesting stuff in that sense. And the idea is that data loaders can replace all of this streamlining code in a project. So your project becomes a lot leaner, and yeah, as I said, less redundant. So let's go through 2 example worlds. So the first one is one where people don't share data loaders, so it's a bit like the status quo essentially. So there's David, who used a previously published dataset in an atlas, but made a mistake when mapping cell types, right? Because some authors had like some name for T cell, and then some authors had some other name for T cell. So there was a need for a mapping dictionary, and there's just a bug in that dictionary. So T cell label switched to B cell label, because nobody ever looks at this code in most cases. Again, after they wrote it. This then actually ended up in a study and nobody ever realized it, right? So this is super bad. Like nobody wants this to happen. Then second case, different place. Felix publishes the data set, but it's not one of these like super flashy, very big, expensive single cell data sets with hundreds of thousands of cells, but it's a smaller one. But it's still, right, like it's a lot of effort to curate these data sets. So now, because people like go through the literature and sort of like try to find a compromise between how much time they spend on curation, given like the impact that that curated data has on their project, they would often choose to not curate these data sets. So even though this is a meaningful data set and maybe like very interesting in terms of like the particular setting that was sampled, right? This data set may not receive as much attention as it could have. And then third example, there's Leander, and he tries to create a cell atlas out of literature data sets, but opts to not include all of the data sets, for example, such as Felix's data set, because it's smaller. And then as a result, right, you have an incomplete picture of what's happening in the literature, and then you may miss things. So 3 different cases. So one, essentially a bug resulting in a wrong result, a study not getting like as much literature community adoption as possible, and then 3 like opportunity costs in the sense of like something interesting was missed because not as much curated data was accessed as would have been possible. Now in a better world, there's John. He publishes a data set under DOI, so it's called a 10A. And then at the same university, there's Aviv, who works on this data loader. So essentially, in contrast to inflating the dark matter of single-cell analysis code, John published this as a Sfaira data loader, right? So now it becomes very easy for Aviv to increment that. So just make a small change and sort of update it to GitHub. So there would be a version 2 of this 10A loader. Now, there's a third person, Sarah, who lives very far away, right? Like she might not even be aware of the research that John and Aviv are doing, but she sees that there's a data loader for dataset 10A, and she's interested in the dataset because it matches the tissue that she's interested in, or like disease they are working on. Now, Sarah can directly use this data loader 10A version 2, right? So get curated data directly. But in this case also, because she's interested in definitions of batches, she may contribute further to this by also annotating batches. So then again, updating the data loader on Sfaira, right? So this 10A data loader sort of like becomes a living thing that evolves with the community and becomes better and better over time. And then lastly, I mean, at this point, this data loader is sort of like perfect in the sense that all metadata that one could care about are really well annotated in there. Fabian enters this field and is looking for data, finds 10A and can use it directly, right? Like that is like really, so Fabian doesn't even like need to look into like how to curate things anymore. He can just like directly use this, which is super nice. And he also won't make mistakes in this black matter part of the code because this is really sort of like community approved curation code. Yeah, so this is essentially where we want to go. Having these very transparent data loaders. And today and tomorrow we'll teach you how to use them. So today is more like how to use them and how to generate them during curation. And then generation is interesting for you because your code becomes more reproducible. You share your efforts, right, which is nice. And if it's your own data, you increase impact because people will be able to access it more easily, and that will just generate more citations. Will also attract more sort of like critical feedback on this data, right? So like there's really like a ton of advantages. So if there's no questions on this session, then I would progress to session 2 now.
We just had one question coming like this second. I'm just going to read it to you.
Yep.
So Tim is
Is it me or is Leander out? Sorry, I didn't hear you. I'm not sure whether I was out or if you were out. Did you see the question, Felix?
Yes, let me look it up. Just a
sorry, I
minute.
think my connection just dropped.
Yeah.
Should I
Do you
read it
want
out
to
again?
read it, Leander? Or should I read it?
I'm just going to read it. Sorry for that. So Tim is asking, does this mean that if you are creating a meta-atlas using multiple data sets and you add it to Sfaira, should you add not only the DAI and the data set of your meta study, but also add the individual data sets to Sfaira?
Yeah, it's a good question. We also we go over a bit more in detail tomorrow, and I think we'll probably even have example cases among the stuff that people will work on. So in brief, there's a data loader for every study essentially, and a meta study is just another study. We have metadata that indicates that this is not primary data and also where these cells are coming from. So essentially, first approximation is, let's say you have a meta study consisting of 2 other studies. So there could be a loader for these 2 studies, and there could be a third loader for this meta study. And then there's metadata that tells you in this third study where this data is coming from, so that you can, for example, in some cases, you want to avoid having duplicate entries of cells in a larger database. So then you might counter select against that. But yeah, in principle, it's just a study. You can, use it there. Cell by gene operates also the same way. So you can upload meta studies to cell by gene the same way you can upload studies. Sort of moving forward, we anticipate it to become a lot easier to build these meta studies because the source data is already curated, right? So right now we're essentially not at this point. We're just collecting the meta studies and studies. But let's say 3 months from now, you're at a meta study and you start your efforts with looking for the curated data sets on Sfaira, then you probably base yours on that, right? But end of the day, you could still, again, write a data loader for that, just to make it clear to people, how you subset the cells, for example, right? There's always going to be some transformation of the data. Yeah.
Thank you very much.
Cool, thanks. Was that it?
No more questions for now, I think.
Yeah, cool, nice. Cool. So, so that was the introduction. So as outlined before, now we have a few short sessions on API chunks. The first one will be on what we call universe and dataset API, which will be a bunch of slides from me now, and then a short tutorial by Leander. And then afterwards, we also have a short break. So just for your coffee planning. So universe and dataset. This is a bunch of classes that we'll go over. The core intuition, and sort of try to associate this with the different APIs because they all have a very defined application case. The core intuition of universe and dataset is that this is the key tool within Sfaira to obtain curated data. So this is your entry point to get and handle curated data. So as a brief preliminary, we'll go over what a data loader is. Data loaders are part of the universe and dataset API, but yeah, again, we'll go over them in a very detailed fashion tomorrow. So I have a diagram of a streamlining process of data here on the right. So in blue, we have sources of data. So there's a raw data provider in blue, right? So that could be Geo. So there's like some unprocessed count matrix on there. Then there's a data loader provider, which is Sfaira GitHub. And then there could also be a curated data provider. So that could, for example, be cellxgene data. Now in orange, and from here on, I use this notation that like this blue is like a remote data location, so a cloud server essentially. Orange is an instance of a class. And then purple and pink are local data. So purple is data on disk, pink is data in memory. So you can download raw data from a raw data provider. So you can download unprocessed count matrices from Geo, and they'll lie somewhere on your laptop's disk, essentially. Now, what Sfaira does is that it instantiates a data set for each of these data sets. So parts of a study, essentially. And this data set, orange, sits around this raw data B now. This data set class, so the instance, has a load function, and it has some metadata that both come from the data loader of B that's hosted on Sfaira. Using this dataset specific stuff, so the data loader, the dataset class can transform the raw data B on disk into a data object in memory, so it can load it into memory, and it can structure it, so it can streamline it. It's sort of like repeating the curation process. So within that one class, you can essentially go into memory and streamline. Tomorrow, we'll go over what this metadata of the data load and the load exactly are. But for now, this is like the core intuition that you need. Now, this orange dataset class is always the same class. The key idea is really that the data loader abstracts everything that's dataset specific away from it, exposes it to you as a curator. And then all of this like really like code intensive and also like performance sensitive stuff is hidden and shared across all of the data sets that there are. So that's the data loader. Now, curation is the process of writing this data loader B stuff, right? So this other stuff is Sfaira backend. You'll not really be exposed to it during the curation. And so during curation, you would take the raw data, download them to the machine that you're working on, write the loader, and then you can produce this curated data B inside of the dataset instance. Now, let's say curation is complete. Either you completed it, somebody else completed it, and you're now interested in accessing a curated object B. So you can use a caged object from a curated data provider like cellxgene. So this is here, right? And this also works within the dataset class. So here, right? So the dataset class allows you to go through the curation workflow. So you use data loader and sort of like fold it over your raw data to get the curated data. But it also allows you to access a remote caching server. So both ways you get curated data. And these are essentially the two options that you have. You either, if your goal is to get streamlined data, so streamlined undata or HFIDs, you can either access them directly or go through the workflow starting from raw data. Now, how does that fit into like the bigger context of working with data on your machine? This is a bit more complicated diagram, but it's also, yeah, it's a nice anchor sort of like to localize all of the components that we will talk about today. So on the right in blue, we again have these three cloud entities. So the three classes of cloud entities that you usually interact with. So raw data provider, Sfaira GitHub that hosts the data specific data loaders, and then the curated data provider.
David?
Yeah.
Just very quickly, we just had a request in the chat. Maybe when those diagrams get a bit more complex, if you could use your cursor to just, you know, as you move through the diagram.
I don't actually see my cursor. Do you guys see it?
I don't know.
Okay. I'll try to mention the components a bit more explicit in this one because it's a bit
perfect.
Weird. Sorry for
Thank
that.
You.
Yeah, so these, right, so in blue, these cloud entities on the right, and now on the top left, we have a universe instance. So, right, the session is universe and dataset API. So on the last slide, we saw what a dataset is. It hosts data loader, which is just a bunch of study-specific code, and it can control essentially how you move a data set into memory and how you streamline it. So here, this universe on the top left has a dataset A instance and a dataset B instance, right? So right now, this is a universe that contains the two studies of this example case. And again, as before, within dataset B, the raw data provider provides the purple raw data B, which can be transformed using functionalities of the dataset class into the pink curated data B. And again, you could use the data, you could use the universe to obtain curated data B directly from a curation provider also. So this is the universe, right? So you see that the universe interacts with all three blue cloud entities and on every data set that is contained in it. So the universe, the idea is essentially, it's a bunch of data sets and it's all of the data sets that you care about in this part of the pipeline. And yeah, in a minute we'll get to like how you arrive at a subsetted universe. The point is though, so this, right, like you'd be in a Python session and you would instantiate a universe and then you'd have that and it would contain a number of these data sets. And you may choose to streamline them, right? So it may contain the raw data or it may contain the curated data. So what do you do with that depending on the application that you have? So one very basic and probably like the most common one would be to just emit undata objects from this universe, right? Because this data set B is already centered on an undata object. So you can just pool all of the undata objects from a universe. Let's say you have like two data sets in there, A and B. You can just get the list of these two objects. Then the second option would be to write all of the streamlined data to disk. And there we have particular store formats that we'll talk about later today. So that's the store API. And yeah, either essentially, so and from the store, you can also emit then a data or something that we'll talk about later, which is a stream. And then this data as an undata or as a stream arrives essentially at your output. So like that could be like a ScanPy workflow. It could be like model fitting in TensorFlow, something like this. Now, universe contains all of this stuff to like make working with these A data objects in memory as easy as possible, right? So like, because you can streamline them in a universe, because it has all of the data loader instructions, you can easily concatenate stuff because you can control mapping raw data sets to the same gene dimensions, for example. Or you can control streamlining of cell type annotations so that you can meaningfully concatenate cell types across data sets. And this is really important, right? So you, imagine you'd have a universe of like 50 instances of data sets in it. This is already like a huge bunch of this like dark matter single cell analysis code. Like if you have to like transform them to something that you concatenate first. So this is all handled by universe. Essentially, single lines to that will do all of this stuff that, yeah, we consider sort of dark matter. Any questions to that?
Yeah, we have another one from Tim. So Tim is asking, as we are kind of choosing count matrices as a starting point for providing data in Sfaira, how do we deal with those technical differences you might have upstream of that? So counting or aligning and also genome versions.
Yeah, this is also something we'll discuss tomorrow because it's part of the curation. Specifically, an example for genome versions. We have capabilities to map gene names between GTF versions. So this is essentially what happens then in the backend. We would like streamline all of the gene names to, like, let's say we have like dataset A with like 2000 genes because it's like some filtered matrix, and then we have dataset B with like 20,000 protein coding genes of ensemble releases 102 and 103. We would map both of them to the same, let's say, protein coding gene space of ensemble 104, whatever you're interested in. And then in that space, we can align them. So align them, meaning we can concatenate them. But these are all direct manipulations on count matrices.
Okay, thanks again, David.
Thanks. Cool. And yeah, and a lot more about this also tomorrow. Then let's move on. So universe, right? So that's like the whole set of things. Now we saw a mention of a dataset class. Now, brief pointer for you to just like be aware of that because you sometimes see it in the code. In principle, there's like 3 main classes that correspond to hierarchies of sets of datasets. So dataset is literally one dataset. So it always maps to one A data. If dataset B, if you trigger emission of an A data object, there's one A data object coming out of it. And there's also like no concatenation or something going on. This is really one count matrix that was loaded from somewhere on disk into memory. And if you have a study where somebody saved like 5 count matrices, then there would be 5 dataset instances for that study. Now, a dataset group is essentially a very shallow wrapper around a dataset. It just groups datasets that belong to one study together. So those 5 would be in one instance of a dataset group. And that dataset group would then be identified by something like a DOI, right? So in cellxgene, that's a collection. But yeah, that's essentially a set of datasets. And there's a bunch of things that you would usually do with sets of datasets, right? Like you would concatenate their datas. You would relay an instruction to streamline data to all of its members, right? It's just like, it's a convenience handle for you. And the same is a dataset supergroup. So that is a set of dataset groups. So for us, for example, all of the datasets managed by Sfaira are in a dataset supergroup. And this dataset supergroup is called universe for simplicity. Similar, there's dataset supergroups on interfaces to curated data provider servers. So for example, there's one interfacing cellxgene that just has all of the collections in cellxgene in it. And so important is that by doing this hierarchically, right, like we're able to relay any instructions essentially that you have to all of the members, and that makes that very lean. So universe with 50 datasets in it, you can map all of the datasets to one feature space with a single line. You can concatenate all of their data with a single line because the stuff is relayed. Yeah, so dataset group around it, and then dataset supergroup around that. And usually if you interact with Sfaira, you would use the universe, which is a particular dataset supergroup, which is specifically collecting all of the loaders that are implemented in Sfaira. Is there questions for that? Good, nice. Yeah, you'll see some of that later too in the tutorial. Now, so there was a bunch of mention that, right, like right now in Sfaira, there's like something like 60 studies curated. You don't always want to load all of them into memory, right? Or like even like download all of the raw files. So a key property of the universe and all of these data set classes is that you can perform operations lazily. So you can instantiate a universe that has entries for every data loader in Sfaira. And you can subset it. So let's say, for example, here, let's start here on the left. So purple again on disk data, we have study A, B to M. And you could imagine that the total set of loaders in Sfaira is even more, right? There could be like N to T in there also. So the universe sort of instantiates every data set that there is, and then it can keep only the ones that you have local, or you can use it to download data that you don't have. Importantly though, right, so this collection of all of the data loaders that are in Sfaira, you can subset it. So here, for example, we subset from data loaders A to M to data loader B. And you see that here, right? So study A, study B, study M, in the after initializing, they're still on disk. So they're still purple. After subsetting, they're also still on disk. So you have an object that represents all of the data that is in Sfaira, which in memory would be something like, I don't know, hundreds of gigabytes. So you shouldn't have that in memory. But you can subset it before you even start doing memory stuff, just because we lazily interface these things. And we essentially, the only thing that we know about these data sets are certain metadata. And this metadata we can use for subsetting. So for example, the subsetting to study B, this could have been a subset to any data set that is from mouse and also from retina. Or it could have been a subset to human and any samples that are also diabetic. And here we really leverage that we use ontologies for metadata, which we'll go over again tomorrow, which essentially relate, for example, how a query to a tissue should look like, right? So there may be samples in there that are like from certain subparts of the lung and samples that are just like labeled lung altogether, sort of. We are able to do like relational reasoning within these ontologies. So if you say lung, you get everything that's in lung. Doesn't matter whether it's like exactly the term lung or like upper airways left low or whatever, like very specific anatomic locations that we might have been able to annotate. So here, so two important things. So subsetting is metadata-based queries that use ontologies, so relational reasoning, and it's lazy. So this is like super nice to work with. Because essentially you don't have to know what's in there, right? You just like start looking for stuff and then you see what you end up with. So here we end up with study B. Now, my query for human diabetic samples definitely was study B. Now I may want to like investigate this a bit more. So I trigger a load on the universe, which is then only loads dataset B into memory. So it becomes pink now after .load. And now I may want to use the curation code that somebody previously compiled for this study B. So to do that, we'll call streamline instructions on this universe, which will then move dataset B in memory to a curated dataset B, still inside of the universe, right? And now we could take it out of the universe. So we could emit the A data, and then you can chuck it into some scan pair workflow, whatever you're interested in. You could again write it to a store so that you have the curated objects lying around there for you to address later. Up to you, right? But yeah, so this is a really powerful workflow, and that it interfaces all of the data loaders, but you don't have to do a lot of stuff. It can do a lot of things lazily, and it can do relational reasoning when you subset. Any questions for that?
Nope, no questions at the moment.
Yeah, this might be, I think, especially this year, it might be a lot right now, but you'll see this also in the tutorial. And yeah, this is really like, it's very powerful and sort of like the key entry into working with Sfaira and data loaders. Yeah, so this is also the end of the introduction of this API. So now Leander is going to take over with a short tutorial on the universe and dataset stuff. I moderate the questions a bit. So just continue to ask in the chat on Slack or here. Felix is also going to move to the breakout room now. So if you have any issues with running some stuff, you can also just like join Felix in the breakout room for a bit and ask him. Yeah, otherwise, feel free to follow Leander while he's executing the code by executing it yourself, but you can also just like look at Leander's screen shares, essentially up to you. Like it's sort of like either tutorial or sort of like guided walkthrough. Thanks.
Perfect. Thanks a lot, David. I'm just gonna post the link to the notebook that I'm gonna run through in the chat now, so that if anyone who wants to follow along interactively can go to that link, download the notebook, and run along. This is, of course, only if you have your Sfaira set up already for the session tomorrow, for example. Otherwise, if not, you can just look at that later and just follow along what I'm going to be sharing now. As David said, there's Felix in the breakout room in case you have any issues. And yeah, we're going to get started straight away. So this is where you can download the notebook, and I'm just going to jump to the other tab where I have it running already. So as David has covered, already. We wanna load some data set from the Sfaira universe using the Sfaira API. And even though the whole theory behind it, what David just went through, might seem a bit complex when you first see it, I'm trying to convince you now that it's actually only quite a few lines of code to get you started. It's super easy to use. And yeah, I'm just gonna run this as we go along. So yeah, let's get right into it. So first of all, I guess most people are familiar with kind of standard Python. I'm just importing Sfaira, some system packages. And then I'm defining 3 directories, which I'm going to be using as my Sfaira data repository on disk, which is this data directory. That's perfectly fine to be empty at the beginning. Metadata directory, that's also empty in the beginning. And an empty directory where Sfaira is going to cache some stuff. So if you're loading data sets again down the line, you'll be saving some time. So I've run the first cell and the second cell already. In the second cell, we're instantiating this Sfaira universe, which David has been describing. And so I'm just instantiating that universe class. I'm now providing the 3 paths that I've defined above. The second and the third one are actually optional. So we could just go with an empty directory as the data directory, and from there move on. So I've instantiated the universe. Now, before I'm going to execute that next cell, I'm just going to run through briefly what I'm doing here. So right now in this DS object here, I have the whole Sfaira universe. So this is data from mouse, data from human, data from all different organs. And what I'm trying to do here now as a demo is I'm subsetting the universe to only human data sets. So basically I'm subsetting the attribute organism to human, which we refer to as Homo sapiens according to the ontology that we're using for the species. And in the next line, I'm subsetting to the organ I, because in this case, I'm interested in single cell RNA-seq data sets from the human eye. And while I'm talking, I'm just going to execute the cell so the loading can run in the background. ds.download. This is exactly what David meant with we're kind of really streamlining the whole process of obtaining the data sets. It's one command which automatically pulls all the human eye data sets from the cloud onto your local disk into that data directory, which we've provided up here. And now that we've downloaded it, we're saying ds.load. Again, just one line to load all those data sets which you've just downloaded into your Python session. And what we're doing under the hood here with the load is basically execute those data loaders, which you're going to be writing tomorrow in the data curation session. So just one command for us here, executing all that loading code, which has to be written once for each data set and contributed to the Sfaira code base. Now that we've loaded that data set, and I can already see here in the output that it is now loading all those different data sets from the human eye. So we have 3 retina data sets loaded into our Python session so far, and it's still running. As a next step, we're running the streamline features function, because right now we have these 3 data sets loaded. We of course need to kind of align their count matrices so that all the genes are in the same place if we want to concatenate those count matrices now and get a uniformed data object back. So streamline features basically make sure that according to some ensemble release, which I've defined here, so I've defined the ensemble release 104, and specifically only the protein coding genes from the ensemble release 104 for human, because that's what I'm interested in right now. I'm making sure that all the genes are in the same order according to that release in the 3 datasets which I've just loaded. So now that my count matrices all look the same in terms of what's where, I also want to streamline the metadata before concatenating those data sets, which I've pulled from different places in the internet. So I just say ds.streamline metadata, and Sfaira makes sure that according to the internal Sfaira schema, which has different metadata fields predefined, which are filled as part of the data loader, and you'll see how to do that tomorrow. We're streamlining the metadata, and basically, so if you're familiar with the data structure, we would be reshuffling and filling columns here in the adata.ops attribute, because that's where our cell-wise metadata sits. And now my cell has finished executing as I've been talking. And you can see my last statement here was to print the ds.adata, which refers, adata here refers to the andata object, which contains all the 3 data sets I've just loaded in a streamlined fashion. And you can see from down here, I've outputted that adata object, which I can now use for my downstream tasks. It has predefined columns and ops. So whenever I load the data sets with Sfaira, I know that I will get these fields as observations. They're always called the same thing, and they are all filled according to the relevant ontology. So when we talk about cell types, for example, those data loaders internal to Sfaira make sure that a macrophage is always called a macrophage, and it's not once called a macrophage with a capital M or a small m. So when you subselect this data object for in your downstream process for, say, for example, macrophages, you won't be missing all those different spelling of the same name. And this is also true for diseases, for the assay that the data was captured with. So it's all streamlined and should be very easy to access because of that. So we now have loaded 44,000 cells from the human eye with basically, I don't know, maybe like 10 lines of code, and it's there ready for use, ready to use for you in your Python session. Now, next step would be to demonstrate this for another organ, where we first instantiate the universe again. We subset to human, we subset to lung, because right now we're caring about human lung data sets. And then we looked, okay, what data sets are actually in Sfaira, which are human from the human lung. So you'll notice we're not loading them straight away. We're looking at the IDs to start with. And this allows us to basically look at the list and select one data set or multiple data sets that we care about. And you already see in the next cell that we are indeed selecting one data set, which will in a second appear here in the dataset list. So just like in the cell above, we were, or in the section above, we were interested in looking at all those human eye datasets which are out there. We're now more interested in a more focused way, checking for a single human lung dataset which is in here. So I got the IDs, and I could see in a kind of harmonized way what those data sets are. So basically, first I see they're human, they're from a lung, which makes sense because that's what I've subsetted for. When they were published, 2018, 2019, 2020, the sequencing protocol, the first author of the publication, as well as the DOI of the publication. And you will already notice, yeah, sure.
We have one question from Hugo. At this point, can I be sure that the data has been processed in the same way? And if not, how would I know? I guess roughly in this channel.
Processed in the sense that kind of up to the count matrix, or maybe you can just clarify that. People can unmute themselves, right? I think so.
So, okay, she's asking about whether it's a count matrix or normalized.
Right.
Yeah, I guess like the entire streamlining process.
Right. So right now, if I'm loading a data set in the way I just showed, I can be sure that I'm loading the most raw version of the counts available, because that's kind of the best practice. That's what we ask people to do.
So
when
then
they
how would you know whether, like, suppose the best version of the raw counts available is the normalized version, right? Then where would you see that?
Right. So each of the data sets has an attribute.
And just because this we changed, this is actually one of the things we improved a bit last week.
Cool.
So we used to annotate a normalization routine, sort of like we used to give it like a string that said it was normalized this in this way. But it became a bit complicated to keep track of like what a particular normalization routine is called. So what we have now is essentially you can assign a metadata that says the count data is here, and this is really only the count data. So this is integers. And also additionally, you can also assign a layer, right? That could be in like layers, in different layers. It could be X in the draw, but you can have a second expression matrix, and you can assign that to normalize. And that can be any normalization that you deem sensible then. So what happens relatively often is that people would supply counts, and then they would contribute some, for example, like batch corrected and normalized data that is like good to like visualize gene expression on a UMAP or something like that. So that would be the processed one then. But essentially you have handles, so you have attributes in the class that point to counts and that point to whatever is processed. And by pulling on them, you will get what you want essentially.
So in the data sets, like in the literal code that Leander's showing, like where is the attribute that tells you per data set whether what you have is normalized or raw or etc.?
He's pulling the main object right now, which is counts if it's available. Otherwise, it's the processed one because it's not everybody supplies counts. This would be part. This would essentially be part of the streamlining call. You could define there like what you want and like what you want to counter select.
So this is like brand new features as of this week, which should make this easier. Do you know from the top of your head, David, the attribute of that class to check, for example, for our Homo sapiens lung parenchyma datasets, which we're loading here, that we could check like live right now what it's like?
So you can, if you pick a dataset instance.
Yeah, so I should have that here.
Then you could check whether layer counts or layer process is set.
Okay, so we check for layer counts, which is just an attribute of my selected dataset. And it tells me that the layer counts is in the X attribute of the data object. So that way I know that I can expect actually raw counts in the X object or in the X layer of the FDN data, and I can be sure of that. Does that clarify the question, Yuga?
Yep, perfect, thanks.
Awesome.
Thanks. There's directly maybe a second question from Christoph. How can I access all possible values for the key organism? I'm not sure what values here are. Christoph, do you want to unmute yourself also?
Well, I guess just what organisms we have in Sfaira, right?
Ah, true. Yeah, true. Yeah.
Cool. Do we have an attribute right now where I can just view the organism? I'm not sure we do, right?
It lies under organism, but it's essentially anything that's an NCBI taxon, which is an ontology. So we'll go over this tomorrow. Like, I mean, that's essentially the letter names of organisms, and these are valid keys. Yeah.
Right. Cool.
So, and then if you specifically care about like what's everything out there, that would be an interaction with the ontology class. We could discuss that tomorrow.
Yeah. So whatever is there in the ontology, and then if I want to know if from that specific organism we have something in Sfaira, I would just instantiate the universe as I've done up here, and I would subset for that organism from the NCBI taxon. And if I then run ds.ids, either I have an empty list, which tells me that there is no data sets from that organism in Sfaira, or I actually get some returns and then I know that we do have data from that organism in Sfaira.
And there's one more question from Stefan following up to US. Apart from keeping track if counts are normalized raw scale, do you also test Sfaira if a data set is already filtered in some way? Many people do only supply filtered data. Yeah, I mean, it actually, like if you really look for unprocessed count matrices, or quite often you would find them. And then, I mean, there would still be cell filtered, which is okay, like if they kicked out like cells that are not really cells. but we would usually see all of the gene data up to the ones that were all 0 anyway. Otherwise, there are some special cases in curation how we would deal with filtered observations, but this is like it's a surprisingly rare case.
And I guess you can sometimes also get this through the cell type annotations, right? When you check for the annotations of the data set that tells you the cell type, we have some special strings for cells which are not a cell, like empty droplets, or which are unknown. So you can also filter those out downstream. If you want to make sure that you only keep the cells which are which have been assigned a cell type by the original authors. Perfect. Do we have any more questions of it, or should we just go
I think we're
through
good
this?
now.
Perfect. Okay. So where do we pick up? Okay, so we've subsetted now for the single data set. And if you remember, I got the ID from the list up here from all the human lung data sets. I subselect the single one. And we already checked for Yuga now if what the layers is for the actual count data in the data object. So it's the X layer, so it's kind of the default layer in the data object. That's great. So we can just work with it out of the box. We can also quickly check if the cell type is, if this data set has cell type annotations with the dot annotated attribute. I just ran this, so it tells me it's true. So we do have cell type annotations in this data set. So now I want to subset my universe to exactly this data set. So that's the only one I have in here, because up here I've just queried that data set as part of the universe. And now I want to make sure I kick out everything from the universe that is not this data set. So I subset the universe just like I did before with the organism key or the organ key. I subset now with the ID key to just that organism, to just that data set. And then I do the same thing as I did in the previous section. I download it. I download the file, the raw file from Geo or wherever it lies for this data set. I load it to my Python session, and I grab the A data from the data set from the universe group, which is this DS object here. Now, one thing to note, you'll see in the previous session, or in the previous section rather, I have used ds.adata. And the ds.adata is kind of the adata which contains everything from the universe. So we're concatenating data sets basically. So I always have to run the streamlined features and the streamlined metadata before I access the data object of the universe. However, what I'm doing down here is I access the data object from this single data set, right? I'm not saying ds.adata, but I'm saying ds.datasets and then that one ID, which is in there still, which gives me the unmodified data. So if I specifically query for the data of the exact data set after running load, I can get the data without any Sfaira metadata or feature harmonization. So this is basically the data that the authors have provided, potentially plus some additional metadata that we have added through the data loader. So this is kind of as untouched data as you can get from Sfaira if you don't care about the harmonization and you just want to see what the authors have put in. And you can already see that we have, for example, a lot more fields in the adata.var, which is the gene-specific metadata. We have a lot more columns here with annotations from the authors, which otherwise would have been removed because they're not part of the standard Sfaira metadata schema. But sometimes we are interested in those, so we can access the data through the dataset as I did here without running the streamline functions before. And then that's what we get. And in contrast to that, we can run the streamline features, the streamline metadata. And now the data object will look a lot leaner in terms of its metadata and will again adhere perfectly to the Sfaira schema. Will have all its metadata annotations converted according to the ontologies, as I mentioned before. So the cell types, the disease, for example, the perturbations which might be in the data set. So these are the different ways of accessing this. And now that I've run streamlined features, streamlined metadata, I could indeed also just access the ds.adata, which would be identical to this call. down here. Right, as this is running, I'm just gonna jump to the last section here, where we're actually doing, we're pulling out data sets across organs. There's nothing stopping you from doing that. So so the first lines you'll be familiar with right now already. So we instantiate the universe, we subset, in this case, to a mouse. And now, instead of providing a single organ to the subset call, we just provide a list of multiple organs. So in this case, we're pulling everything lung and liver from mouse that is in Sfaira. We say download, as we did before. We say ds.load to load all those into our Python session, basically executing those data loaders in the background. And then again, as before, we run streamline features to that specific ensemble release. We streamline the metadata, so the cell-wise metadata according to the Sfaira schema, and then we print the data sets again. So as this is running, you can just check the previous cell because that's finished executing now. And if you compare the two outputs here, as I described, the data object after streamlining the metadata and the features has very different column names for your ops and the vars, and also in the unsorted annotations. And you can now be certain that anything you find in these metadata columns will actually be harmonized according to our to the ontologies we've applied to all these metadata. So as you can guess, there's quite a lot of code actually running in the background when you execute those functions, but that's basically what Sfaira is all about. It's making data curation and data loading as easy as possible for you, the end user. And so it's for you, it's two lines of code, and you're probably running like 500 to 1,000 lines of code in the background in order to get all those metadata aligned. Now, we're already seeing we're loading quite a few data sets here. So this is mouse, lung, and liver. We're probably like at 25 data sets here, which are automatically loaded. And then the metadata and the features are streamlined, and then we're outputting the finally data object, which you can then again use downstream in your workflow to test your new embedding model, to put your newly obtained mouse RNA-seq data into context by running some integration approach, whatever it is that you brings you forward with your research. Perfect. While this is finishing executing, David, do we have any more questions, or is that basically it?
There's a few that ask why there's blood data sets being loaded right now. So I think the ones that we've seen the outstream, the SCOG.
Yes. So there is actually a couple, if I'm not mistaken, this is from the cell, what is it called? Human cell landscape dataset from the Google lab.
I think the mouse one, but yeah, exactly. Yeah.
It's true, the mouse landscape, cell landscape. And there we have multiple organs basically in one, in each of the data sets. So hence, sometimes the name in the data set does not exactly match what you're loading.
So maybe like this ID that you're seeing here, this is somewhat human readable, but this is not the metadata that we have, like a much more complex set of metadata that we structure this on. This is, you will see tomorrow why files are called like this, but they would essentially be partially loaded here to only yield what you want.
Exactly. So only like the lung and liver cells from that data set are loaded here right now. Perfect, and we're done. So that's nearly 68,000 cells, mouse, lung, and liver that you can now downstream use. Again, you see exactly the same metadata columns as you saw for the human data before, all harmonized according to the ontologies. Right, so that's it tutorial-wise for the Universe API. Any more questions, David?
I think we're good.
Perfect. Awesome. Okay. I guess we're a bit behind schedule right now. What you proposed of it in terms of when should we
I think we can
continue with the next?
Yeah, I think we can do like maybe like a 5-minute break now. So we just reconvene at 35 and everybody can just like take a short break from looking at the screen. Yeah, but we should be fine. We'll catch up to the schedule in the next sessions. Cool. Yeah, thanks everybody for interacting. And thanks, Leander, for the tutorial. So yeah, next session will be, so it will be a block of 2 sessions, which will be the cell by gene API and then the store API, both accompanied by a tutorial. And then after that, again, a short break. So yeah, we'll just see you again in 5 minutes.
Awesome. Super excited.
Okay, I think we can slowly start again. If anybody has any, can somebody please once say that they can hear me? Just because I don't see anything.
I can hear you.
Yes, we can hear you.
Yeah, awesome. Nice. Cool. So any remaining questions regarding the last sessions, just, yeah, feel free to message us privately also. I mean, right, some of these are probably also legit feature requests, and you could also just post them on GitHub. But yeah, just feel free to contact us. The code that Leander went through, so the Sfaira tutorial is on Sfaira tutorials. And yeah, exactly, like we don't need to share it because the link to the tutorial is already in the agenda. So if you check back to that, you can go over that in your own time also again, sometime when you need it. Similar for the other tutorials that are coming up now. Cool. Then, if you want to leave feedback for any of this stuff, you can also do that on the Google Forms. Yeah, as you prefer. Now, the next session is on the cellxgene API. So cellxgene is a data curator, and was already mentioned before as a curated data cloud server, right? So there we make like a categorical distinction between servers that we declare raw data servers, where people tend to deposit unstructured data upon publication of a data set or a manuscript. And then there's curated data servers, which is actually, I mean, there's hardly any, and cellxgene is sort of like the dominant one, that provide data that is already curated according to a particular schema. So a particular set of rules that define how metadata items are called, which ontology they're subject to. A lot of the stuff we'll go over tomorrow. So tomorrow we also talk about how once you created a data set in Sfaira, you can actually contribute it to cellxgene, right? So pushing data to cellxgene. And now this session today, because this session today is sort of like centered on using data, we talk about how we pull data from cellxgene. So this interface here is part of the Universe and Dataset API that allows you to interact with cellxgene data, much like you interact with local data through like a Sfaira universe, just as Leander described just now. So yeah, the normal workflow is, and that is reflective of what you just saw in the tutorial. You get raw data from Geo or raw data from Fischer, depending on where it lies. Then you get a data loader from Sfaira and a universe instance, sort of like folds them together and gives you curated objects as you sort of like instructed to. Now, entities like cellxgene, they directly host files that are curated according to a particular schema. Now, in this Sfaira data, we have a front end for objects hosted on cellxgene that goes through the download interface by cellxgene. So in that setting, there can essentially be somebody who uses data loads from Sfaira and raw data from Geo to create an H5ID to upload that to cellxgene, right? So that could, for example, be the initial curator or like the author of the study that has a strong interest in publishing a data set to cellxgene. Now you as a secondary user then have the choice of running the streamlining again from raw data locally. That may, for example, be the case, or maybe desirable if you want to update a data loader because you think there's something wrong or missing in it. Or secondly, you have the option to just directly pull the curated object from cellxgene, both through Sfaira Data API. Now, this API goes through a class that's called Dataset Supergroup Databases. So it already has this Dataset Supergroup part in it, right? So we talked about this before. Dataset Supergroup is a group of collections or group of studies. And it interfaces, for example, all of the Sfaira data loaders. So that's the universe. Or this one here, also a supergroup, interfaces all of the database data sets. And here, so cellxgene is the only database that you interface right now. So here, this one interfaces everything that's in cellxgene. So now you can essentially interact with that the same way that you also interact with the universe, and it has all of the same advantages. So it's lazy, but now lazy means, so before lazy meant, right, you have a ton of like raw data on disk and you don't want to load all of them into memory. You want to like subset first, so like figure out what you want, then load into memory. So here you have a ton of data on a remote and you don't want to download all of them. So again, you can subset lazily by just interfacing this like group of data sets where you have basic knowledge of some of the metadata. So for example, I could subset for collection ID, which is sort of like the DY equivalent in cellxgene. Then I could specifically download only this collection, load that into memory, and then again access the data objects in that. Do the same workflows that Leander just described for universe, but here now on this dataset supergroup databases. So very simple workflow, only 5 lines of code, but you immediately have your object. And now it's essentially like depending on the case, right? Like you could choose which way you go, like go via raw data or go via this. Depends also like what's available on cellxgene. What do you want to sort of like map towards to? Maybe you have a very rare streamlined output like format that you need to satisfy, and then maybe it's better to go by raw data. This like really depends now, but you have the options. And importantly also, right, like a lot of this like subsetting loading code, like it looks exactly the same. So like you could also like switch relatively easily. So yeah, we'll do a short tutorial on the cellxgene API now. Again, as a Jupyter notebook, so similar to the one before. Unless there's any questions now before we do the tutorial. Okay. You're still muted.
Thank you, David. So in the exact same directory.
And now I think the sound doesn't work.
You can't hear me.
I think it's filtering out your voice for some reason.
Is this better?
Yeah, no, it was.
Is this better?
Yeah.
Perfect. Okay. So exactly in the same directory on GitHub where you found the previous tutorial notebook, you can also find the cellxgene one. It's called cellxgene_download. If you interactively want to go along. So just as before, I'm importing the standard Sfaira and some other libraries. I define the path where we save those data sets which are in the cellxgene repository. And as David has shown, this is literally all the code you need. I'm just going to go through it and show you some more bits and pieces about it. But this is literally what it takes to pull those a data object from cellxgene and have them ready to use in your Python session. So just like before, where we were instantiating the universe, we're now instantiating the dataset supergroup database because we're specifically only interested in the cellxgene datasets right now. So we're only taking the database group here. We provide the path, and then once we've instantiated that dataset supergroup, we can just call the show summary function, and we see all the datasets which are in there on cellxgene, which are basically available to us to pull with another 3 lines of code. And if we're interested in some more metadata aspects of each of these datasets, we can also, for example, just use a simple Python list comprehension like here, where we go through all the datasets in this dataset group here, and we query a specific attribute. So right now we're querying the collection ID, but you can also be querying the organ or the disease condition, the protocol, whatever you'd be interested in. And it just gets outputted here. And we see right now all the collection IDs which are available in cellxgene for us. So now we pick one of them, and you can use those collection IDs to also directly go to the cellxgene website by basically appending it to this URL, as we've done here. You can click on that, and I jump to the cellxgene website where I have a nice explanation of the data set. I can also manually download them here. So these IDs are basically the universal identifiers used by cellxgene, and they're the same in Sfaira and on the website. So now I subset to that single target collection here. This is mainly because to keep kind of the download in the demo light, you can of course supply 10 or 20 IDs here if you're interested in those, or subset for, again, organ or organism, as we've done before in the universe tutorial. So we've subsetted for that one target collection here. And we can check how many data sets are in this target collection, and it's indeed 2. So this paper had 2 data sets associated with it, and we can now query them individually. So just like before, we hit .download on this dataset group object to pull the whole thing to disk. And then we can actually check on disk with os.list here whether in the cache path, which we're using as kind of our local repository for this data, if the collection has arrived. And indeed, we see we have now a folder on disk, which is named after this collection, which has these data sets inside. Nice. So, and now it's basically exactly the same as in the universe tutorial. We load the data. So we see we're loading these 2 data sets to disk, and then we're outputting these 2 data sets with adata.ls. So before, in the previous tutorial, we were using .adata because we wanted one concatenated one. In this case, as an example, we're pulling them separately into our Python session as kind of a list of 2 adata objects, as you can see here. So this is exactly what you can then use downstream. But instead of pulling them both at the same time, you can of course also access an individual one with the dataset ID and work with that instead. As we go on, there's another use case here, which is as shown in the previous tutorial, you might not want to subset directly with the collection ID from cellxgene, but you might be interested in any data set that is from lung, for example, human and mouse in this case, because that's what we have lung data for on the cellxgene database. So we pull everything lung from there, instantiate the supergroup subset just as we did previously for organ, and then we show the summary, and then basically based on this dataset supergroup object here, we can then output the data as we did above, or further process them as we wish. So that's it from the cellxgene tutorial. Feel free to pull that from GitHub, play around yourself, and see what's there in the cellxgene database that might be useful for you.
Thanks. There was one question from Rob. What data set exactly maps to in terms of like sequencing layer and stuff? So I think this is also become a lot clearer tomorrow, like what the different cases are, but it's essentially always a count matrix that was deposited somewhere. So in these cases, right, you saw 4 data sets there, so that essentially corresponds to 4 count matrices, which usually, right, like it depends on data reporting and stuff, but usually that would be one sample of a single cell experiment, for example, or it could be in some cases, it's like a bunch of concatenated samples that were analyzed together. But really the defining, like the object defining characteristic is that this is a published Cone matrix. So it makes sense for us to like deal with that as is, because often there's a bunch of metadata shared across this matrix. If there's cell-specific metadata, it's usually annotated for all of the cells. So this is why we interact with it this way.
Nice.
Cool, I think, yeah, it was good. So that was the cellxgene interface. As mentioned, in parallel to Universe, essentially, to manipulating raw data on disk. Now, there is a second sort of like pillar of interacting with data, which is the store API. And this pillar is essentially characterized by the philosophy that stores close the gap between curated data and streamlined models under the Sfaira models API. So this is the key thing. This has a lot to do with access of data, both performance of access, but also modes of access. And this API is store classes, card classes, and then there's essentially data chunks. And we're in this session, we go over that. So yeah, looking back to where these stores come from, you'd have a universe or any dataset group, dataset supergroup instance. And these all have the ability to write streamlined data sets. So here, for example, curated dataset B in pink, once it's in memory, to a store. So a store is just a location on disk. So here, right in purple, this is curated dataset A and B. They're both lying in the same directory on disk. And now we can instantiate a store class on top of that directory, and it will give us access to the curated datasets within there. So why would we want that? So I mean, there's the obvious cases that include you have curated some datasets and you just want to save the H5 IDs and maybe like individually send them to some collaborators, or you're just interested in using them downstream and you don't always want to go through Universe. So there you'd naturally usually directly work with H5 IDs and then emit end data from that. So rightly so, right? You can also here in this diagram, the store in the bottom left, there is an error to an A data in memory. So you can just load it into memory and then the store is essentially just a bunch of H5 ID or other storage format on disk. But there's a second case in which you wouldn't want to load entire objects into memory, but in which you would just want to stream content. And this is what we'll talk a lot about today. So yeah, let's imagine you have a store with 10 data sets. So here A, B, C, D until 10th letter of the alphabet. What would you do with that? So you could load these 10 objects into memory. That may work if they're small enough, right? And then you may concatenate them such that they become one really big data. And then you could push that into a machine learning library, for example, and use it like as training data. Similarly, you could also just stream from all of these objects, treat all of them as training data on disk, and then just like load into memory what you need at any given point in training, which is usually a mini batch. So this is something you typically do if you work with models that use stochastic optimization like TensorFlow and Torch, for example. And this is essentially then, yeah, where a bunch of store functionalities come in. Importantly though, right, like remember, like the store always comes from a universe. So you would also in that case, or in that application scenario, you'd start with the universe to build the store, and then you start. So the setting we're faced with now is that we have very big universes, right? So there's like millions and millions of cells in these universes. And I mean, we can subset them, but in principle, we could say we just write all of them to a store. And then we have this one directory with like a huge amount of curated data in it. And now you may have like multiple modeling projects in which you access this store. And these models, they would like, I don't know, like model like certain characteristics on all cells, or they would like model like cells of a particular tissue, only stuff like this. So essentially you have this store on disk, which is going to be like hundreds of gigabytes large. And there's a whole bunch of stuff in it. And it's like so much that, I mean, like realistically, you don't really know what's in there, right? Because I mean, you don't have a collection or recollection of the metadata of all of the studies in the store, because yeah, I mean, right now it's like more than 50, and then soon it's going to be more than like 100. So yeah, so there's a whole bunch of data there, and you don't really know what's in there, but you know what your models want, right? So it may be that you have a model that only wants lung data or something. So here, the first concept that turns out as really important with dealing with that regime of data is that you're able to do metadata-based subsetting. So in a store, I want all cells from lung. This is a really important query and something that's really often useful. Similar for all of the metadata, right? So this is something that a store enables, and it allows you to do that again lazily. Like remember, like something that universes are really good at is like doing lazy operations as long as possible. Again, the store is the same. So you can subset a store instance to only the cells that belong to the metadata partition that you're interested in. Next, models under the Sfaira model API, they parameterize what they want. So like in terms of like input feature space, in terms of metadata. And this is something that you can directly interface into a store. So there's sort of like a seamless transition from data in a store to data in a model. So here the important concept is, so I want to be able to subset metadata to decide what training data is, for example, right? Because that defines what a model can learn. And otherwise, I just want to let the model pull out of the store whatever it needs to. I don't want to define all of these transformations myself. So yeah, this stores also do. And then lastly, I mean, most of the time you want to do stochastic optimization. So the standard in any machine learning framework, which will require you to load mini batches from disk, because you don't want to have all of the data and store in your memory. And for this, you need data representations on disk that are able to emit batches quickly. So these are data representations like ZAR or Parquet in Python. This is representations that we use to write stores. We have like one representation of single cell data that we call distributed access optimized. So you see that as DAO throughout the code. That is particularly fast at access. Yeah, we have like a whole bunch of stuff like research and benchmarks going on around how you can pull mini batches as quick as possible from such a very large collection if you know what you're looking for and you have like some knowledge of like what your access trade-offs are. That even then goes to having these stores like lie around on cloud servers and stuff. So yeah, so if you're in a setting that looks like this, right? So like very large collection, you don't really know what's in there, but you probably need to do metadata-based subsetting. You want to directly sort of like interface a model into the data collection and then stream fast from disk. Then you can use the store, and then you can like actually like train models on like huge amounts of cells without like a ton of memory. So like if your model is small enough, you can actually like really train this like on a pretty small machine. Any questions to that?
No questions so far.
Good. Now, let's revisit the interface from universe to store once more. So now that you know sort of like what we use the store for, one important paradigm in high performance computing is sort of like accessing only the data that you need to access and sort of like defining the data in a way that the amount of transformations you have to do on the fly is minimal. So one strength of universe is that it can write stores, so it can write streamlined data of like a target format to a store. So let's say, for example, you know that you're gonna fit models that expect only the protein coding genes in human genome. Now, it would be wasteful to have a store that has all of the genes in there and maybe like in a bad ordering, because then when you pull the mini batches, you'd have to reorder and you essentially just use a lot of CPU time from that. So here, essentially, if you really want to set up like a high performance machine learning pipeline, you'd use the universe to write a store that is specific to your application. And then you use the high performance access capabilities of stores. And here, right, like usually this universe writing something that is specific to your scenario, this is a huge effort usually, because I mean, you'd have like hundreds of these individual data sets on your hands, and then you'd have to make sure that they all have the same gene space and that their metadata sort of align. Like this is a huge effort if you don't interface the collection of data sets that you have through like one instance that then just like relays the instructions that you have. So here it's really like, if you think about like large projects from a high performance computing point of view, it's really important to like cleverly use the universe to write what you need and then exploit the store. Now let's look a bit more into the store. You'll also see that in the tutorial. So the store sits on a data collection. So again, as before, it's lazy in the sense that it doesn't load the data into memory. You can subset the store with functions that look very much like the subsetting of a universe. And at some point, you'll end up with a selection of cells. Let's say you have a store of like 10 million cells. You'll end up with a selection of cells that you're interested in. You can, so now there's like different store backends. So that could just be HF5ID files, or it could be these like Zara Parquet centered DAO formats. Doesn't really matter what the backend is. You can check out a certain selection of a store into a card. So that's a method on the store class, and it gives you an instance of a card, which is still lazy. So this is a selection of the store that's now just specialized into providing you with its contents, which are particular data packages, right? So mostly this is gonna be expression data and then observation as metadata. So it's a very reduced set of things you would want to stream, right? Like there's no concept of unstructured in streaming anymore. There's at this point then no concept of var anymore, because the var is already defined by the interface. So this is going to give you this subset of var that you care about. So this is usually X and ops is like the two things that you need. You can emit them into an data, obviously, and then chuck it into an any of the frameworks that use data. Or you can just stream batches of X and ops directly into a machine learning framework. And there, so the card essentially facilitates that. It doesn't really matter like what the backend looks like, it's gonna enable that. And one example, so let's say you have a store that's written in DAO, then your count data or your expression data is gonna be represented as Zara on disk, which is a block representation of count data. And your observation metadata is gonna be represented as Parquet. Now the card interfaces the Zara arrays with Dask, which allows you to lazily pull chunks of the data to disk and perform transformations on them. So for example, you may have a model that uses like the total counts of the cell as a size vector or something like this. So there you could just like write a transformation of the data that yields the expression vector of the cell itself plus the sum of the expression vector. But importantly, right, this is a simple transformation that you can do if you have X and ops, and we'll go over that also in the tutorial. So yeah, remember store, subset, checkout, and then you have a card, and then the card gives you a stream or it gives you a data set. Now, the stores are pretty flexible, so you can do different things now. So first of all, you could like concatenate different stores, and also different directories that are in different parts like of the hard drives that you work on. So this is the scattered case here on the left, where we have A and B together in one store, and then later somebody else wrote a C store. Maybe A and B were like on a cluster drive and the C is on your local drive. now you could get a card with A, B, and C in it, and then just like stream observations of the 3. Then there's a distributed case where C is maybe on some cloud server, like a S3, for example. Now you could just like build a mixed store onto your local AP and the remote C, and again, stream observations from the pool of ABC, right? So the card gives you, the card really like pulls all of the bits and pieces together that you need to just like pull data according to a certain like emission schedule based on the data selection that you have. And then the last case would be, this may be an interesting one. Let's say you have stores where there's multiple organisms inside of them. So A and B are human, C is mouse. Now they're not on the same feature space anymore, right? Because the GTFs are different. So there's a different number of protein coding genes, for example, between human and mouse store. They both can be interfaced with an individual store. So here on the right, the store human and the store mouse. And you can also chuck them both together in a store multi, which from a user interaction point of view looks very similar to you. It also emits a card, but this card now allows you to, for example, emit human and mouse observation in turns, or put like batches with both of them inside, like essentially do stuff where you can deal with observations that come from different feature spaces. And again, right, all of this is like access optimized. You can do metadata subsetting on it, whatever you feel like. One point on building the stores. So I said before that the main route is probably going to be for you to write them from a universe instance, which means that you need a data loader for it. So that thing needs to be curated in Sfaira. There's a couple of cases where you may want to like execute a model, for example, on data that is not streamed in Sfaira yet, and it's not even very desirable in the application case. In those cases, you can interactively chuck a dataset into a specific dataset class by only defining like a bunch of very core metadata attributes, and then push that object that is streamlined in memory into a specific store, which you then can again like extract a card from and plug into your pipeline. So I think this is like too much of a workflow for today, but just keep in mind, you can also use this pipeline if your data set is not curated in Sfaira. There's like a way, and the key steps along the way are essentially a class called dataset interactive, and then store a data. Just keep it in mind, and you can go back to the code documentation when you need it. Yeah, so that is the store API. We'll run a small tutorial on this also, which I'm gonna share, and Felix is gonna present.
Yeah, so thanks, David, for sharing. So I'm shortly gonna walk you like through like some examples for the store API. And so like the first part, like if you want to, you can execute like the notebook cells on your own, but like be aware that especially like the downloading takes a while to run. So might also be good if you just like follow along, and maybe if you're interested in, then execute it like afterwards. So like the first part is kind of like downloading the data. So this is already executed in our case. So if you want to do it yourself, I think it takes like 5 or 10 minutes to execute all the cells. And then we come like to the second part, which is like creating our store. So like when downloading the data, this is kind of like at the moment or stored like in an inefficient way, or inefficient in a way that it's like very costly to access like random bits of your data. So what we are doing here, we are writing like this DAO store to disk, which is kind of like writing the ops data in Parquet format. And especially what's like the most important is like writing the gene expression data, because like that's like the vast majority of your data, or like the biggest chunk of your data, in like set array format. And like the set array format kind of enables you to randomly access like parts of your data, and while being very efficient, that you don't have to load like the whole data set into memory when you want to access like just parts of the data. So kind of like there, we are like iterating out over all of the single data sets we have downloaded, and kind of like load them into memory, and then write them in a like efficient way to disk, that we can later on, like when you want to do like random batch access, that we can like efficiently sample like cells from there. So did you already run this, David? So maybe you can,
myself. Yeah, this is already run. Yeah.
You can like just execute it, and now we're coming like to the interesting part. And maybe like one more thing to the store API that's quite costly, writing the store. And the cool thing is kind of like if you have new data sets, it's very easy to do this. You don't have to rewrite the stores for everything. You just have to rewrite the stores like for the new data set you added. So that's very easy, like to add new data if you like want to expand, have like another interesting data set which you want to add to your analysis. You don't have to do this part like all over again. And now we come like to the part of like to the interactive part of the tutorial. So like, first of all, like our goal is kind of like we want to load the data to like train a model like to do cell type classification. So for this, we kind of like want to have the gene expression as our input, and like then want to kind of like predict the cell type for each like input gene expression. So kind of like the ops data we want to have like is we want to have like the cell type column, like so the cell type annotation for each cell. So that's kind of like what we define here, like all the ops data we want to load. Then in the second part, we kind of like define our data store, or like our handle in Python, which we use like to read from the data store, which we just wrote like in the cell above. So that's kind of like we just point where's the store, which format is it, and like which columns we want to load. So if you execute this, then it is very fast because it's like all done lazily. So we don't load like the whole data set into memory. So that's all very nice. And then we can see like in the next cell, like we have one store like for mouse data and one store for human data. And kind of like for our tutorial, we just want to look at the human data. So we kind of like just, in this case, just select like the store for like human. In this case, it's like Homo sapiens. So that's kind of, now we have our store instance. And now as the next part, we kind of want to create a card to actually look at the data we have. So what we do there, we execute like or called store.checkout to get a card. So if you run this, we have our card, and now we're calling card.x. This is kind of like the big gene expression data matrix. We can see like for the tutorial, it's quite small data set. So we have like 4,500 cells and like almost 20,000 genes. Then we can also take a look at the ops data, which in our case just has shape 1, because at the beginning we selected, we just want to have like the ops data for the cell type. So this ops data, just like you can see down here, it's just a normal pandas data frame, contains like the cell type annotation for each sample we have in our store. And now we come like to the generators, which we then would use like kind of like to train a TensorFlow or PyTorch model. So, and the cool thing there is like, first of all, we kind of like, or the first thing we want to do like to train our like cell type classifier is like we have string labels, and we kind of like want to one-hot encode this like to actually train a classifier on, because like we need like a numeric vector and cannot just like predict like the string labels. So what we do here is just like some code like to do some one-hot encoding of the cell type labels. So I think that's not so interesting for the tutorial, and the interesting part now comes now. Kind of like, because like for the store, it's kind of hard to, or like I like to say like this, like we want to have just the raw data on disk, and like in this what we call like map function call, we kind of like define what how the output of the generator should look like. So as the input to this map function, we have like the X sample, which is like corresponds to the gene expression, and like the ops sample, which is like just some random chunk of the ops data. So what we have, we kind of like, because like the X sample is a desk array. So what we do there is just like to actually do computing on it. We just call numpy as array on it to kind of like call dot compute on the desk array, that we have like a numpy representation of the batch we are currently looking at. And then we also want to output like the cell type. And for this, we just have like our string labels, and we want hot encode them, and then we return them like as a tuple. Like the first one corresponds like to the input, which is in our case like the gene expression, and also have the cell type. And the cool thing like with this map function is it can output like whatever you want. If you would have like an autoencoder architecture, we would just like output like the gene expression twice to make this like really modifiable, and you can do like all kinds of manipulation there to really make it suit to like the output you want to have from your generator. So then we actually, in the next cell, we get like the generator we want to have. So for this, we can select like the indices which we want to look at. If you set it to none, it will just like iterate or like sample from the whole data set. But for example, which is like uncommented here, we could also just sample from the first 100 cells in our case. So this would also be like if you want to have like a train test split or whatever. Then we specify like the map function we want to use. We define this here in the cell above. And we also have to specify like which ops keys are there. So again, in our case, it's just the cell type. And then in the next section, we can just generate like a normal Python generator from it. So for this, we call like the .adapter method on it, and specify we want like a normal Python generator, which is like I said, just a normal Python generator. And then we also specify that we want to repeat our data set 5 times. We can also set this like to a negative value, and that it would repeat like forever. And then we can just call like the next method or like next from the generator to get like one sample, which in our case is like the gene expression array in the first part, and then like the one hot cell type encoding like in the second part. Then we also have support like for PyTorch data loaders and tfdata.dataset. And first of all, if you look like at the PyTorch data loader, kind of it just creates like a PyTorch data loader, which you can then easily load and like use to kind of to train your PyTorch models. For this, it's just like the normal PyTorch data loader, and you can pass down like all the keyword arguments relevant to the PyTorch data loader, just get passed down to the constructor, and it returns your PyTorch data loader, which in this case like returns PyTorch tensors corresponding to like the map function we just have. And very similarly for the tfdata.dataset. So for this, it's just calls kind of like under the hood, it calls tfdata from generator from our Python generator function. And then we just get like the TensorFlow dataset output. And kind of like for there, you can, as it's a TensorFlow dataset, you can call .batch on it, you can call a custom map function on it, and so forth. So in this case, we just want to have batches of size 128, and that's what we're getting there. Yeah, so that's kind of like how you would use like those generators to, for example, feed like your data into like a TensorFlow or PyTorch model. Yeah, so that's it from the for the tutorial. I hope I was able to give you a bit of an insight how you can use like the distributed format like to efficiently train models.
Cool, thanks, Felix. Yeah, again, as before, if you have any questions about that, just you can also write to us separately. There's a bunch of new stuff also coming for the stores. Yeah, just essentially watch this space if you're interested in that. Yeah, and this concludes the second block for today. I would suggest we do a 5-minute break again, and then reconvene at 4:25 CT for the last bit, which will be managing private data And then any of you who might have open questions regarding choosing data sets tomorrow or installation stuff, that would also be then. And it looks like we'll be roughly on schedule again. Nice. Yeah, so we'll see you in 4 minutes at 4:25. Okay, we start again in like half a minute. We start the screen share already.
All right, should we get started?
Yes, let's do it.
Perfect. All right, so good news first. We're 3 slides from the end of today's slide deck. So that's everyone in the audience 3 slides away from mastering day 1 of the Sfaira workshop, with of course some Q&A afterwards if you still have some questions for our session of tomorrow. But yeah, final 3 slides, very simple message, managing private data. So we anticipate that quite a few of the use cases of our Sfaira package will be that you as researchers want to put your labs, proprietary data, private data in context of some public data sets which are out there from, for example, the same species, same organ, through a data integration or some other quantitative comparison. Now, we understand that it might not be suitable for you in order to be able to, load your labs data conveniently with Sfaira to submit public data loaders to GitHub and let everyone else see what data you have and how it's loaded. So there's the use case of, I have some private data locally that I want to load, and I want to kind of use it in the same workflow together with the Sfaira data. So what to do about that? Now, there are basically 2 ways. So the Git and GitHub savvy people in the audience will have already thought about, well, I can just clone Sfaira or create a fork and then clone it and have my local copy of it, maybe do an editable install with pip, and then I can just add my data loader, write my data loader using the Sfaira CLI as we'll do tomorrow, or manually, add that to the directory structure where it belongs, and just use it as if it was part of Sfaira without submitting it. Of course, from what I just said, you can already tell that that's maybe not the most convenient option. And that also might create some issues downstream when you at some point want to update Sfaira because there was some cool feature release that you want to use. And then you have to kind of pull from Git, make sure you don't submit your changes. And while that's perfectly doable, it's definitely not convenient. So I want to quickly present to you a second option, which is basically a simple, quite lightweight extension package to Sfaira, which happily is called Sfaira extension, which we've developed, which allows you to add data loaders to Sfaira and in a local way. So you clone the Sfaira extension package, have it locally, and add your data loader to that one without having to touch your Sfaira installation. So you can basically update your Sfaira and just use it, treat it as a normal pip package, and manage your local data loaders in the Sfaira extension package. So as you can see here, basically from the directory structure of Sfaira extension, this is just a screenshot from GitHub. This is basically exactly the same as the directory structure in Sfaira. And it's not just the directory structure, which is the same. It's basically the whole way of how you write the data loader and you put it in there. So basically writing a data loader for Sfaira extension and keeping it locally is exactly the same workflow as writing a data loader for Sfaira and submitting it through a pull request to the public, which means you can very conveniently load and provide your local data within your lab. And should you at some point publish your data in hopefully a very high impact journal, you can then just take those data loader files, copy paste them into a normal Sfaira installation, create a pull request, and give it to the public. So there's literally no difference between a data loader that sits on your computer locally in terms of its structure compared to one that sits in the cloud. So, and the lightweight Sfaira extension package basically allows you to manage these local ones separately without having to touch your Sfaira installation and then potentially running into installation issues, or having to deal with Git commands every time you want to update Sfaira. So that's it in terms of managing local data with Sfaira alongside the public one in a seamless way.